Okay, so let's get started. So hello everyone. Welcome to our today's Zoom seminar. Uh, it's we have a big treat today. Uh, it's really my pleasure to welcome our today's speaker, Dr. Sun Shaocong from Beijing, to offer us a lecture. So I think a lot of audience, including myself, actually learned a lot of previous T-cell literature from Shaocong's lab. But it's really my pleasure to have Shaocong today on our platform. So, so Shaocong currently is a distinguished investigator and director of Institute for Immunology in the Chinese Institutes for Medical Research in Beijing. So, at the same time, he also served as a chair professor in the Capital Medical University of China. So, Shaocong received his undergraduate degree from Hebei University and a PhD degree from Stockholm University in Sweden. Then he pursued his postdoc training at the Gladstone Institute of Biology and Immunology in UCSF. So from 1994, he joined Penn State University as assistant professor, started his independent research program, and then subsequently went through the rank to become a distinguished professor. And then from 2007, Shaocong moved to University of Texas MD Anderson Cancer Center, where he served as a professor and deputy chair in the Department of Immunology, and director of the Center for Inflammation and Cancer, and hold the Mohsen Tapas endorsed chair in immunology. 2024, he moved back to China and joined CIMR. So, as I mentioned, a lot of us probably already being very, very familiar with Shaocong's work. His lab actually mainly studies signal transduction in immune and inflammatory responses with a focus on T cell responses. His lab really made a number of major contribution to the T cell biology field, including pioneer work in NF-kappa B signaling pathway and finding regards the role of ubiquitination in immune regulation. He was the one of the very first lab to demonstrate IK beta inducible degradation as the fundamental mechanism of NF-kappa B activation. And his lab pioneered the discovery of non-canonic NF-kappa B pathway and demonstrate of its regulatory machinery. Um, at the same time, his lab also made numerous findings regarding to how ubiquitination regulate T cell function in autoimmune disease and anti-tumor immunity. More recently, his lab had made an important contribution to the understanding of signaling machineries regulating anti-cancer T cell responses. And very important part of his ongoing research is to dissect the signaling network regulating T cell activation, effector function, and exhaustion in cancer immunity. Uh, well, as I said, probably a lot of us has read different literature from Shaocong's lab. His lab published more than over 170 research papers. And then I think the important thing, not only he actually being the lab has been extremely productive, he also have a very Souchong has a very strong record in training young scientists. He has trained more than 40 postdoc and then nearly 20 PhD students, and a lot of them actually now are very active in both academia and the industry. With all these achievements, he has received a number of honors, which I'm gonna not gonna list of them, but just a few example, including AAS Fellow, NIH Merit Award, MD Anderson Cancer Center, Living Legacy Legend Faculty Achievement Award, and a lot of the list just go on. So again, it's absolutely my pleasure to have Xiaochong on our platform. Um, Please go ahead. We are very much looking forward to your talk.
All right, thank you, Chuan, for the nice introduction. It's really a great pleasure to present here and meet with friends online. For those of you who are not familiar with CIMR, this is a relatively new research institution located in the main campus of Capital Medical University in Beijing. So if you know any friends interested in working in Beijing, please recommend them to apply to us. This is a just wonderful place to work in. All right, as Chuan introduced, we are studying T cells mainly in both autoimmunity and cancer immunity. And today I'm going to talk about our work in the cancer immunity project. All right, so I know this audience doesn't really need an introduction, just want to be very brief to get started. Then we know that for acute, let me get the laser. So for the immune responses against acute infections, we know that the generated effector T cells, along with innate immune cells, could eliminate the pathogen in a timely manner to terminate the primary immune response and generate memory T cells. But for immune responses against a chronic infection and cancer, the immune system could not eliminate the pathogen or cancer cells, leading to persistent antigen stimulation. And under such conditions, T cells differentiate towards to a dysfunctional state called exhaustion. And these cells, basically, they reduce or lose their effector function and proliferate capacity, and they express high levels of inhibitory receptors known as immune checkpoints. So our focus is to try to understand the signaling mechanisms that regulate CD8 T cell function and exhaustion, and particularly the process of this differentiation, intra-tumor differentiation process. So we are particularly interested in the signal transduction mediated by the TNF receptor superfamily members. And these receptors are quite interesting. They are induced along with T cell activation and serve as co-stimulatory receptors that are required for sustained T cell activation. So we all know, we're familiar with how T cell get started for the activation. But the process actually also requires the TNF receptors for sustained activation and effector function. So even though these receptors are mainly studied in acute infection models, there's strong evidence that they are also involved in anti-cancer immunity. A postdoc in the lab, Hongping Wu, performed a bioinformatic analysis of murine melanoma single-cell RNA-seq data set, and identified 12 clusters of CD4 and CD8 positive T cells. I just want to point out the 2 clusters. So the terminal exhausted CD8 T cells, as you know, they have really much reduced effector function, and consistently they express high level of the immune checkpoint genes, and very low level of the co-stimulatory genes belong to the TNF receptor family. But the progenitor exhausted CD8 T cells express a high level of the genes including TNFRs, as well as effector molecules like interferon gamma perforin, and also some level of immune checkpoints. And so these cells are actually very important. They mediate, they play an important role in anti-cancer immunity, and also respond to immunotherapies, especially the immune checkpoint blockade therapy that you know right now is widely used in the clinic to treat advanced cancers. And this is, of course, based on the pioneer work of Jim Allison and Tetsuo Honjo. And the idea to block the interaction of these inhibitory receptors with their ligand and thereby release the function of T cells in anti-cancer immunity. But the other way to boost the anti-cancer immune response of the T cells is to target these co-stimulator receptors, and most of which belong to the TNF receptor superfamily. So this is to use agonist antibody to stimulate them. Therefore, it's called hitting the gas pedal approach. This approach has been explored in both clinical trials and preclinical studies and generated fairly promising results, but also a lot of challenges. For example, clinically, the response rate is very low and with a severe adverse effect, especially such as the inflammatory liver toxicity. So we believe that these problems at least are partially due to the complexity of the TNF receptor signaling pathways. It's known that the TNFRs target a number of pathways, including the MAP kinase and the canonical NLRP3 pathways that are actually known to mediate inflammatory responses, and they are likely contributing to the adverse effect of the therapy. And these pathways also respond to the various other immune receptors. But as Chuan introduced quite a few years ago, we pioneered the discovery of a different signaling pathway known as the non-canonical NLRP3 pathway. And this pathway is highly specific. It's almost exclusively responding to the TNFRs. And a major step is the proteasome processing of an NLRP3 precursor protein called P100. And this protein contains in the N-terminus the NLRP3 subunit PP2 and the C-terminus an ICAPB-like structure. So it makes the P100 a super ICAPB-like molecule that blocks the nuclear translocation of NLRP3 members, especially RelB. So the signal-induced P100 processing involved the degradation of the C-terminus, which lead to the generation of PP2 and nuclear translocation of RelB. And we further demonstrated that this pathway is critically dependent on the NLRP3-inducing kinase NIC. Initially, NIC were thought to be a kinase that regulates the canonical NLRP3 pathway, but what we found were that the NIC mutation in mice block the processing of P100, but without affecting the activation of the canonical NLRP3. So it's a specific regulator of this pathway. And what's interesting is that the NIC could integrate signals from different TNFRs. And this makes NIC a quite attractive therapeutic target. Another question is NIC pathway really required for anti-tumor function of the T cells, and whether NIC activation is sufficient to promote anti-tumor T cell responses without involving the other TNF signaling pathways. So we generated T cell conditional NIC knockout mice and used two syngeneic tumor models, the melanoma and the colon cancer model. And in both models, the NIC deletion in T cells could severely affect anti-cancer immunity, showing by the increased tumor burden and decreased tumor infiltrating CD4, CD8 T cells, as well as the effector CD8 T cells. So what this suggests that the NIC is indeed required for anti-cancer immunity. It has a T cell intrinsic function. But the next question is, can we somehow manipulate NIC to boost anti-cancer immunity? In this regard, we have previously shown that NIC is a constitutively active kinase. It's regulated by a very interesting mechanism through the protein ubiquitin degradation pathway. So under steady conditions, when NIC is de novo synthesized, is quickly bound by TRAP3. TRAP3 target NIC to a ubiquitin ligase, including CIP, TRAP2, etc. So this will keep the level of NIC extremely low to prevent its signaling. This is how NIC is controlled. Now, the activation of NIC involves the degradation of TRAP3 that lead to stabilization and accumulation of NIC through de novo protein synthesis. So this means we can try to mimic the receptor-induced NIC activation through an inducible transgenic model using the CRE-ER model. So in this model, we could induce NIC expression in the tumor-bearing mice by injecting the mice with tamoxifen. And so you can see that if we do this without activating other TNF receptor-induced signaling pathways, it is sufficient to drastically reduce tumor burden and improve the mouse survival. And this is associated with a drastic increase in the intratumor T cells as well as effector T cells. So this suggests that NIC is a very potent anti-tumor immune regulator. So to assess the therapeutic potential, we performed cellular adoptive cell therapy using the PML1 TCR transgenic mouse model. As you know, these mice produce CD8 positive T cells expressing a transgenic TCR specific for the melanoma tumor antigen, GP100. And by generating the crossing with NIC mice, we generated wild type and NIC transgenic PML1 T cells and use these T cells to treat the melanoma tumor bearing mice. And so compare with the wild type T cells, the NIC transgenic T cells displayed much stronger anti-tumor function and intratumor survival or expansion. So we don't know how NIC regulate the T cell, whether it's expansion or survival, but the intratumor level is much higher than the wild type T cells upon transfer. And also they contain higher level of effector T cells. So currently we are expanding these studies using the CAR-T therapy model and see whether the manipulating NIC might be approach to improve CAR-T cancer therapy. Now the question is, how does NIC regulate the anti-tumor T cell response? So obvious question is, does NIC regulate the T cell exhaustion? Because we know T cell exhaustion is a major problem in anti-cancer immunity. So we get on the exhausted T cells based on their expression of the immune checkpoint molecules. And you can see that the expression does not really inhibit T cell exhaustion. In fact, the absolute number of the exhausted T cells in the tumor of NIC transgenic mice is much higher than in the wild type tumor. This is due to the overall increase of T cell number in the tumor of the transgenic mice. So this made us wonder whether these cells are really functionally exhausted or not. And we look at the production of interferon gamma and find that the wild type T cells indeed produce very little interferon gamma, suggesting an exhaustion phenotype. But the NIC expressing T cells produce interferon gamma very abundantly, and they just behave really like effector T cells, no exhaustion phenotype. Furthermore, we performed a single-cell RNA-seq analysis and found that the NIC expression causes the expansion of CD8 positive T cells express the effector signature genes. So overall, the studies suggest that if we express NIC, somehow it can preserve the effector function of T cells displaying the exhaustion markers. All right, the T cell exhaustion is regulated by different mechanisms, but one major mechanism is inhibition of metabolism. Under normal conditions, when T cells are activated, they rapidly shift their metabolism toward to glycolysis and also further increase the level of oxidative phosphorylation. And this is very important for maintaining the effector function of T cells. But in tumor microenvironment, the metabolism is severely attenuated due to different mechanisms, such as inhibition by the immune checkpoint PD-1. So we performed a seahorse assay and found that when we isolate the wild-type T cells from the BCTing tumor, there's still pretty good level of glycolysis and oxidative phosphorylation, but the NIC deficient T cells were almost completely impaired in these metabolic activities. So this is very striking. It suggests that NIC deficiency may render effector T cells more vulnerable to metabolic suppression in the TME. And this probably contribute to the exhaustion. In other words, the NIC probably maintain the metabolic fitness of T cells in tumor microenvironment. Now we know that a major signaling pathway that regulate T cell metabolism is the PI3 kinase AKT mTOR pathway. And so we look at this pathway and find that actually NIC deficiency has no effect on the activation of this pathway, as shown by the normal phosphorylation of AKT and the mTOR substrates. And so we also look at the expression of a large number of genes associated with glycolysis and no difference at the RNA level. So quite surprising. But when we look at the protein level, we find a major phenotype on the expression of HK2. Even though the RNA level, no difference, the protein level has a huge difference. And so HK2 is a rate-limiting enzyme in the glycolysis pathway. And it is strongly induced along with T cell activation and correlated with the induction of glycolysis. But this induction is defective in the NIC deficient mice and T cells. And if we express NIC transgenically, the transgenic T cells have a higher level of HK2 expression. We also actually look at the tumor infiltrating T cells and shown same thing. If we knock out NIC, the HK2 expression is reduced. If we express NIC transgenically, you further promote the expression of HK2. So obviously, NIC has a very important role in controlling the steady level of or inducible level of HK2. But now the question is HK2 important for the NIC function? And so we cross the HK2 flux mice with the inducible NIC transgenic mice. Now, if we inject the mice with tamoxifen, it not only induces NIC depression, but also knock out HK2. So as expected, if we express NIC transgenically, it reduces tumor burden, increase the tumor infiltrating T cells, and as well as effector T cells, okay? But what is really remarkable is that if we knock out HK2 at the same time in these T cells, it largely, although not completely, reverse the phenotype, increase tumor burden, reduce the number of tumor infiltrating T cells and effector T cells. So this suggests that HK2 plays an important role downstream of NIC in the regulation of anti-cancer T cell responses. Then previous studies suggest that if you constitutively express NIC in T cells using the CD4 crate, as you know, this starts from early on, from the developmental stages of T cells. Somehow, the overexpression of NIC could cause autoimmunity, include the severe body weight loss and the tissue inflammation. And so in the lung and the colon, we were able to reproduce this data. You can see that from the body weight, you can tell that the NIC transgenic mice have severe body weight loss and severe inflammation in the colon and the lung. But the point here that if you delete HK2 in the T cell conditional transgenic mice or NIC, it completely rescued the phenotype. Now the body weight is restored, inflammation is inhibited. So this suggests that HK2 not only regulate NIC-mediated anti-cancer immunity, but also regulate autoimmunity, suggesting that HK2 really is a major target of NIC. Okay, now the question is, how does NIC stabilize HK2? And we did a bunch of testing experiment and found that if we incubate the cells with lysosomal inhibitors, it restored the expression of HK2, but not restored by the proteasome inhibitor MD132. And so the lysosomal degradation is promoted by autophagy, as you know. So autophagy is a process that is dependent on several core proteins, including the ATG proteins, one of which is ATG5. Now, it is remarkable when we delete ATG in the NIC T cell conditional transgenic mice, it completely rescued HK2. And this clearly tells us that NIC regulate autophagic degradation of HK2. And this, in terms of the additional mechanism, we know that autophagy respond to stress signals, including oxidative stress, like a ROS level. You can see that NIC deficiency greatly enhanced the level of ROS in both naive and active T cells. So actually, in the beginning, we were very confused, like why the naive T cells also have this. Now, I want to remind you that the NIC does not respond to the TCR signal. NIC responds to TNF receptor signal. So under homeostatic conditions, there are TNFRs on the T cells that already could trigger a certain level of NIC activation, even though it's low. You already primed the T cells for TCR response, okay? So this is very important to remember. And even in T cell, tumor infiltrating T cells, the ROS level is very high in the NIC knockout T cells. More importantly, if we incubate the cells with a ROS inhibitor, it restores the HK2 expression. So the conclusion here is that NIC prevent autophagic degradation of HK2 by controlling cellular ROS level. So I'm going to skip the rest of the data, just give you a summary with this cartoon. We have previously shown that NIC target the non-canonical NCAP pathway that mediate effector T cell generation and survival. The current work suggests that NIC also target T cell metabolism. I didn't show you the data. We have a mouse model that block the P100 processing without evolving the early stages. We have a P100 mutant mice. So that mice does not give this phenotype, suggesting that this pathway is NCAP independent. So this pathway is critical for maintaining T cell metabolism and preventing exhaustion. I didn't show you the data. NIC works through direct phosphorylation and activation of a metabolic kinase called G6PD. And this kinase catalyzes the pentose phosphate pathway. And important function of this pathway is generation of NADPH required for conversion of glutathione from oxidized to reduced form that control the intracellular ROS level. So when there's no NIC, you obviously have accumulation of ROS because of lack of NADPH, and ROS then mediate autophagic degradation of HK2 to block the glycolysis pathway. And that makes the T cells vulnerable for metabolic deficiency and exhaustion in tumor microenvironment. Okay, we know that T cell activation requires T cell receptor signal and co-stimulatory signal, But now it's clear it also requires cytokine signal and nutrient signal. And in the following, I'm going to tell you a story about the regulation of cytokine signal, particularly the regulation of IL-15. So among the cytokines, IL-2 and IL-15 could induce the activation effector function of CD8 T cells and NK cells. These two cytokines actually they share the IL-2 receptor beta gamma receptor heterodimer, but they have their own alpha subunit receptor. So this make their function kind of quite different. Because the trimeric receptor IL-2 is expressed on Treg cells, actually very high level. One problem with IL-2 mediated immunotherapy is the induction of Treg expansion and causes immunosuppression. But IL-15 does not have this problem. So IL-15 has been shown to induce strong anti-tumor immune response. Actually, last year, a drug called N803, which is IL-15 receptor alpha complex, has been approved by the FDA for the treatment of bladder cancer in combination with BCG. Now, of course, IL-15 therapy still has some issues, some technical issues, because the IL-15 is not stable. And IL-15, high dose IL-15 can induce severe inflammation. And so it's quite hard to control. That means it's still very important to understand how the signaling triggered by IL-15 is regulated. And this actually has been done by a number of laboratories. We now know that the IL-15 induces three major signaling pathways, one of which is the PI3 kinase AKT pathway. Remember, this pathway regulates T cell metabolism and effector function. So I'm going to tell you a story about the regulation of AKT induced by IL-15 through this molecule called OTV1. And this is a deubiquitinase. As Tran introduced, a long-standing interest in my laboratory is to study the protein ubiquitination. So the ubiquitination is mediated by ubiquitin conjugating enzymes and counter-regulated by the deubiquitinases. We have been mainly studying the OTO subfamily because it's association with regulation of NK-B and immune response. For example, Averma, Vishwa Dixit, and others have shown that A20 is an inhibitor of canonical NK-B and inflammation. We have identified the CIMR as an inhibitor of the non-canonical NK-B pathway in B cells. We also find the TRABIT as an epigenetic regulator of two important inflammatory cytokines, IL-12, IL-23, in the induction of inflammatory T cell responses. So in our cancer project, we performed a bioinformatic analysis of human database. In this case, the human skin cutaneous melanoma sequencing database. We found that the OTV1 expression is inversely correlated with expression of CD8 effector gene signature. And this is actually very obvious. And it is also inversely correlated with patient survival. And so this suggests that OTV1 somehow probably negatively regulate anti-cancer immunity, although we were not sure whether it functions in immune cells or in tumor cells. Nevertheless, the bioinformatic analysis showed that the OTV1 is expressed in different populations of T cells and enriched in the exhausted T cells and Treg cells. And so, furthermore, the T cell conditional knockout of OTV1 could, let's see, there are some technical issues here. Okay. So when we delete OTP1 using a CD4 crate in T cells, we found that it causes homeostatic activation of CD8 T cells, as shown by the huge increase in the percentage of memory-like CD8 T cells and decrease in the naive T cells. And when we saw the naive T cells and stimulate in vitro, you can see that the CD8 T cells are from the knockout mice were hyper responsive to TCR CD28 stimulation. But this phenotype is not seen in CD4 positive T cells. And this is quite surprising because OT1 is expressed abundantly in both CD4 and CD8 T cells. And so this took us a long time to figure out the mechanism. And I'm going to tell you later on. But first of all, through RNA sequence analysis, we found that OT1 deletion mainly promote the expression of genes associated with metabolism and effector function. Actually, downregulate some of the immune checkpoint genes. So by CIMR analysis, we confirmed that deletion of OT1 T cells indeed promote glycolysis. Now, just to make a long story short, for the sake of time, we perform a lot of studies and find that actually OT1, even though the OT1 knockout T cells hyper responsive to TCR CD28 stimulation, if you directly look at the signaling using in vitro deletion method, the OT1 does not directly regulate TCR signaling. It actually regulate IL-15 induced AKT activation. And when we deleted the OT1, you got a strong AKT signaling that promote actually or prime the T cells for this metabolic response. So here's a piece of data. You can see that if you knock out OTP1, it does not affect the activation of STAT5, but it strongly promote the phosphorylation of AKT. And mechanistically, the IL-15 induces the AKT ubiquitination, which facilitate the binding of AKT to the membrane phospholipid PIP3. And this is required for AKT activation on the membrane by upstream kinases. And this step is catalyzed by the ubiquitin conjugating enzyme UB13. It's a KC3 type of ubiquitination and negatively regulated by OTP1. So this is how it works. Now, more important question is how does IL-15 signaling promote TCR CD28 stimulated T cell activation? So this is, people have been talking a lot that IL-15 sensitize TCR signaling, but we perform an experiment, try to find out what's going on here. So what we did was to perform a mixed T cell transfer. We use the OT1 model. This one, because this just allow us to isolate a lot of naive CD8 T cells. So then we transfer the T cells into either a wild type recipient mice or IL-15 receptor deficient recipient mice. And after 7 days, we isolate this transfer the T cells to look at their priming effect. So first of all, we did in vitro stimulation to see their response to the TCR CD28 signal. You can see in these mice that have IL-15 receptor signaling, that OTP1 deletion greatly enhanced induction of several cytokines by the TCR CD28 signal. But if you isolate the T cells from these mice that have no IL-15 receptor signaling, and you no longer see this effect, okay? And the basal level of some of these cytokines is also lower. So this suggests that the IL-15 prime T cells for response to TCR CD28 stimulation, which is negatively controlled by OTP1, all right? Now, to do one more step, we look at how it primed it. We isolate the T cells freshly, and immediately prepare RNA for RNA-seq and RT-PCR analysis. So RNA-seq suggests the induction of a bunch of genes that we're repeating with confirming with the RT-PCR shown here. You can see under the homeostatic conditions, a lot of these genes involved in effector memory and the stem cell signatures are already expressed, and they are hyper expressed when you knock out OTP1. Okay, all of these genes is like this. Now, if you prepare T cells from the IL-15 receptor deficient mice, you no longer see this. That's over here. Okay, you no longer see this. This is very clearly that IL-15 receptor prime CD8 T cells for TCR response through promoting homeostatic expression of genes involved in memory and effector function or stem cell signature. Now, so this explains the previous finding that the IL-15 lower the threshold of CD8 T cell activation. Now we know that OTP1 deletion robustly enhance T cell stimulatory function of IL-15. And the question is, how does IL-15, OTP1 regulate autoimmunity and anti-cancer immunity? So our prediction is that the OTP1 ablation may be pertinent to the autoimmunity and anti-cancer immune responses. So we employed a model. You know that one of the CD8 T cell mediated autoimmunity is vitiligo. Vitiligo is that the CD8 T cells kill melanocytes. So under normal conditions, of course, the CD8 T cells are tolerant to the self-antigens expressed in the melanocyte. But there's an interesting phenomenon. That is, some melanoma patients undergoing immunotherapy display the vitiligo phenotype. And this turned out to be a good phenotype. Those patients who display vitiligo phenotype turn to have a better cancer treatment response. So this is actually, if you think about it, it's not hard to explain because melanoma are derived from melanocytes. They share antigens. Patients with stronger melanoma-specific T cell response will also kill some melanocytes. So that's an interesting phenomenon. We employed an animal model of vitiligo. This is again the PML1 mice. Remember, these mice produce CD8 positive T cells that respond to GP100. GP100 is a common antigen of melanoma and melanocytes. But under normal conditions, these mice don't develop autoimmunity because T cells are tolerant to these self-antigens. They're okay. Only when they are very old, they display a little bit phenotype. But when we cross with OTP1, we find a very clear phenotype. And the wild-type mice don't develop vitiligo at least until at least 9 months of age, have no phenotype. But if you delete OTP1 T cells, the mice develop vitiligo as early as 3 months of age. So we don't have to do genotyping. If we look at the code color, we'll know which one the knockout, which is the wild type PML1 mice. So when we look at the T cells in the lymphoid organ of these mice, we find that these wild type mice, of course, are mainly in a naive state. But the T cells from the knockout display more than 50% are CD44 high, meaning they are already activated. And so when we did the in vitro stimulation, re-stimulation, we find that these T cells actually, they don't respond to the ovalbumin, which is a non-specific antigen. They strongly respond to the GP100, confirming that these cells are already activated by the self-antigen GP100. This is interesting. This suggests that OTP1 is crucial for CD8 T cell immune tolerance. You know that tolerance regulators such as PD-1 and CD4 serve as important target for cancer immunotherapy. So we ask now, does OTP1 deletion actually promote anti-cancer T cell responses? And the answer is yes. Now here we use inducible deletion model because if you remember that, actually I probably forgot to mention that OTP1 not only regulates CD8 T cells, but also NK cells. I didn't show you the data. So in this case, we want to do an inducible knockout model that includes both T cells and NK cells. This is a whole body knockout. In a way, it kind of a mimic pharmaceutical inhibition of OTP1, right? So we did this, we find that if we delete OTP1 inducibly, it greatly reduced tumor burden and increase tumor infiltration with both T cells and NK cells, as well as a population of dendritic cells, CDC1. As you know, CDC1 are characterized by the strong ability to cross-present to activate CD8 T cells by tumor antigen. It's a very important population of cells. Now, by using the antibody-mediated cell depletion assays, we find that the depletion of either NK cells or CD8 T cells could largely reverse the phenotype. You can see that the inducible knockout control the tumor growth very well. Now, if you deplete NK cells or CD8 T cells, tumor growth return to the wild type mouse level or even higher. And in terms of mechanism, the NK cells not only function as killer cells, they actually have a strong regulatory function. If you deplete NK cells, it completely inhibit the tumor infiltration with CDC1s. So this is very remarkable. And this is probably because the function of NK cells to recruit CDCs into the tumor. So anyway, now in terms of therapeutic potential of OTP1, we performed animal model CAR T therapy model. So as you know that for CAR T therapy, it involves isolation of T cells from patient blood and do in vitro retroviral or other genomic manipulation to express chimeric antigen receptor specific to the tumor cells. And then we can infuse the T cells back to the patient to kill the tumor cells. So the CAR T, as you know, has been working very nicely in the clinic to treat blood cancers, but they are so far not good in treating solid tumors. And so the antigens used in the FDA-approved CAR T therapy include CD19, BCMA. They are both expressed on B cells and B cell leukemia, lymphoma cells. So what we are trying to do is to generate a solid tumor model, solid tumor model expressing this CD19 as a surrogate antigen. And we also generated wild type or OTP1 deficient CAR T cells and use them to treat the tumor-bearing mice. And so the result turned out to be quite striking. You can see that the wild type CAR T cells work to a certain extent, as reported by many others. It is not really effective in killing the melanoma tumor model. But if you delete OTP1, you now see it has shows much stronger and much more sustained effect in suppressing tumor growth, and also dramatically extended the survival of the mice. So we are trying to do more translational studies and hoping to be able to see some effect also in the clinical setting. Now, to summarize in the second part, the deubiquitinase OTP1 negatively regulate AKT axis of IL-15 receptor signaling in both CD8 T cells and NK cells. I didn't show the data on NK cells. It worked beautifully also. IL-15 sensitized CD8 T cells for antigen-stimulated activation and function, which is controlled by OTP1. So we think that the OT1 deficiency in mice profoundly could inhibit, enhance anti-tumor immunity through unleashing the activity of both CD8 T cells and NK cells. Okay, and so this is demonstrated through both directly using the knockout mice and through adoptive transfer studies. Finally, we show that if we delete OTP1 in CAR T cells, it promotes CAR T therapy, at least in mouse model. And of course, we need to do more work to see whether this also functions in human T cells. So the work I talked about was actually performed by former postdoctoral fellows, including Mei Gu, Xiaofei Zhou, and some collaborators. So this is my current group. Actually, we need to update the picture. This is the initial group in the laboratory. And I also want to thank our collaborators. Thank you for your attention.